I'd like to welcome to you interactions between constituent parts 4A4. I've already covered a lot of this stuff, so I'm going to kind of fly through some sections. Talked about digestion and the enzymes and hormones that are involved in that. Talked about the gastrin, which is going to be stimulated by protein release into the stomach, and how it's going to cause then the release of the acid and the digestive enzyme pepsin. And the path by which it does it is by clicking on the CCK receptor on an ECL cell. That is going to be releasing a histamine. The histamine binds to an H2R receptor on the parietal cell, and that's going to cause release of the acid. And of course the stimulus is protein. Secretin is going to be one of the most important hormones, and it's going to be causing bicarbonate to be released from the pancreas, as we discussed in class. Bicarbonate is going to lower the pH of the acidic chyme that's coming from the stomach into the small intestine. It stops the activity of pepsin and activates all the digestive enzymes. This is straight from your book. There's not a whole lot here that you're going to need to know for the test, other than probably the salivary amylase for digestion of carbohydrates, pepsin for digestion of proteins, and lipase for the digestion of fats. And the structure here, just be kind of familiar with the fact that the liver, where the bile is manufactured, and the pancreas, which is going to be releasing the bicarbonate, the digestive enzymes, they're all going to join together at the pancreatic duct right here. And a little bit better picture showing the chyme coming in, the location of the liver, and the bile duct, or the pancreatic duct. This picture you definitely need to know, so you want to see the mechanism of the CCK, what it does, the secretin, what it does, and of course gastrin, which we've already mentioned. Remember that the bile is going to be pumped from the liver into the small intestine, and that the enzymes and bicarbonate are going to be pumped from the pancreas into the small intestine. So here is a description. Spend as much time as you like on that to look that over. And again, a little bit more detail here. Alright, so we're going to talk about blood pressure and blood flow, and we're going to be talking about blood vessels and how they affect the gas exchange. So in humans we have a closed circulatory system, which means everything is encased in a blood vessel, and we have three types of blood vessels: artery, capillary, and vein. Arteries break into smaller things called arterioles, and then capillaries are the smallest. That's the site of gas exchange, and that's what we're really focusing on in this section here, is how the gas exchange is going to be occurring at the capillary beds. And then venules return to veins, which return to the heart. So here is the circulation here, and you can follow the pathway if you like, but remember, focus on the capillary beds here in the body and the capillary beds here in the lung, where the gas exchange is going to be occurring. A couple of differences here for you in the blood vessel structure. Remember, arteries and veins have sealed so that no blood comes out of them, and when blood does come out of them, you have a serious issue, more serious from an artery than from a vein. And you can see the thick wall of the artery, and the vein is thinner. You can see that in the picture up here. It's a thinner wall, and it does not have the same muscular surrounding structure to it. The endothelium right here is going to be in the inner lining of all of our blood vessels, arteries, capillaries, and veins, but it's most important in the capillaries. They pretty much just have a very thin layered membrane called a basal lamina, and then the endothelium. And that's all there is. And capillaries, by contrast, arteries and veins are leaky. Stuff can't seep through them. Not blood cells, but the constituents of blood, the plasma. And what happens is that blood flow coming in here is coming in at very high pressure, and that is because the arteries are going to be carrying high pressure due to the pumping action of the heart and the pumping action of those muscles that surround the blood vessel. And that is going to force fluid out. And that fluid that's going to go out is going to contain all of our nutrients and our oxygen. And then on the other hand, as we move through here, the pressure decreases, and now the pressure outside is greater than the pressure inside, as you can see from the graph down here. And so we get fluid flowing into the capillaries. That's interstitial fluid that's containing waste products and carbon dioxide. So the gas exchange that's occurring at the capillaries is occurring because we have high pressure coming in, which forces the blood plasma out carrying the nutrients. Then as that flows out, so much of it flows out that the pressure is actually higher on the outside, and it's going to flow back in on the back end of the capillary joining the vein, and that's going to bring in our waste products like carbon dioxide and metabolic waste products. And here's a bigger diagram of the same thing, and the same thing just for a little bit better vision. Alright, so we can either have air or water for our respiratory exchange. The less oxygen is available in water than in air, and it takes a lot more to get water from oxygen from water, which is why you have organisms like fish having to have constant breathing, because they have fewer oxygen atoms, and they have a more difficult time getting them out. And therefore we have to have large gas exchange surfaces, and ours is going to be gigantic. If you unrolled your lungs, I think you could probably cover a tennis court with the size of the surface area. So in our brain, the medulla is going to regulate the control center here, and then there are going to be sensors that are going to be coming in from the lungs and from the blood that are going to be reporting on changes in the carbon dioxide concentration. As the pH decreases, as the carbon dioxide concentration increases, the pH is going to decrease. As the pH decreases, these blood sensors in the carotid arteries are going to send a signal to the brain. So if you imagine yourself running really fast, you're going to have a large production of carbon dioxide. That carbon dioxide will be dumped into the blood. That carbon dioxide will bind with the blood. We'll see that in a few minutes, and form an acid, carbonic acid, and that's going to lower your pH. Lower the pH of the blood is going to stimulate signals in the brain, in the medulla and pons, to cause you to breathe faster and your heart beat faster. So lots and lots of oxygen, carbon dioxide. And here again, we're going to be coming into the lungs. We've got low partial pressure of oxygen. I'll show you this in just a second. And we have a basically a diffusion gradient, a concentration gradient. So if you look at the picture here, if we are going into the lungs, the partial pressure of oxygen is very low, and then coming out, the partial pressure of the oxygen is very high. Carbon dioxide is high, and it is lower outside the lungs. So some of the carbon dioxide gets dumped, and lots of oxygen gets picked up. Not all the carbon dioxide gets dumped, but some of it does. So carbon dioxide transport to the lungs is based on this carbonic acid up here that is going to be binding to the iron atoms in the hemoglobin, and then it's going to diffuse out as carbon dioxide into the airspace in the lungs. And in the tissue, the carbon dioxide is going to be coming high here, and it is going to be then diffusing into the blood vessels, so that the hemoglobin's going to pick it up. So over here, the hemoglobin is going to be releasing the carbon dioxide, and over here, the hemoglobin's going to pick it up. So hemoglobin can pick up lots of oxygen, and each of our blood cells has thousands of hemoglobin molecules. And what we see in this little curve here is that as the partial pressure of oxygen decreases, more oxygen is going to be unloaded from the cells into the tissue, right? And as the pH changes, so if we have a decrease in pH, we even have a stronger dissociation curve. So as the carbon dioxide goes into the blood and lowers the pH by making it more acidic, it is going to change the dissociation curve so that the pH, or the hemoglobin, is going to release more oxygen into the cells to try and reduce the carbon dioxide concentration and increase the pH. So here's just a little bit of information about what we just talked about. All right, now we're on to plant transport. and plant transport is going to be two-way movement. In the bottom here we have oxygen and water, and other minerals that are be picked up from the roots, and they're going to be transported all the way up to the top of the tree through the leaves. And then we have the leaf production of nutrients such as sugar through capturing a light energy that needs to be delivered to all the cells of the plant that are away from there. So in this one we have two types of diffusion, or two types of movement, a short distance and then a long distance. One of them is active and the other one is passive. And depending on the property of the plant tissue and the property of the cells, or the molecules. So, but selective permeability is something we've talked about a lot, and we see it here again. Okay. So most solutes going to go through these transport proteins that will be in the membrane, and what they are going to do is they're going to cause what's called a proton pump. And this proton pump is very similar to what we saw in the nucleus, or in the nervous system with the potassium and sodium leak channels and the ATP, or the sodium potassium pump. And they're going to create what we saw again, very familiar, the membrane potential. So this is going to be pumping protons out of the membrane where they are already high, and that's going to make this area over here more acidic, and that is going to allow stuff to diffuse in into the plant cell. So here we go. Here's a transport protein for cations, and then a co-transport of anions with hydrogen. So the hydrogen is going to come back in, and as it does come back in, it's going to be bringing in a nitrate. And then we may even have a neutral solute that we're bringing in through this co-transport. So once we pump this stuff out by active transport, it's going to travel back through, and as it travels back through, it is going to be bringing other stuff with it. So osmosis is going to be the primary reason how we can control water uptake and loss. And we're going to be measuring something called water potential, and that's going to tell us which direction the water is going to move, and it's always going to be moving from a higher potential to a lower potential. So here is the symbol for water potential. We do have some calculations based on that to go over, and it's measured in megapascals. So a water potential of pure water at sea level and room temperature is 0 megapascals. So pressure and solute concentration are going to affect the solute concentration, or solute potential. And solute potential is called the osmotic potential. And it's got this little subscript here. Please pay attention to that. Okay. And then we have two other kinds of pressure here for you too. So here we have water potential, and it's going to be moving from a high water potential to a low water potential. So if these two started out even and you added solute over here, then that means it's going to have a decreased solute potential, water potential here, and a larger water potential here. So it's going to move from a higher to a lower water potential. Okay, so water potential is going to be have a huge impact on how well plants can pick up water. So if you took a flaccid cell and placed it where there was a higher solute concentration, what you're going to see is that the cell is going to shrink, and then it is going to plasmalize, which means it's going to kind of break down. Right? On the other hand, if you put it in one with a low water concentration, or high water concentration, a low solute concentration, it's going to swell up. So this is why we get wilting in plants, and that of course happens to us every time we fail to water our plants for a few days. They're going to go flaccid, and then they'll go turgid. And of course, if you do that several times, the plants will no longer be able to maintain homeostasis, and they will probably die. So if you have managed to kill plants through inconsistent watering, that is what you've done. So the proteins that are involved in moving water in the plant are called aquaporins, kind of an obvious name. And then It's basically how often we can phosphorylate these proteins, and of course every time you phosphorylate a protein, you have basically added a phosphate to it from ATP, which means that this could be an energy requiring reaction. So we have three traveling routes for the water. The first one is called the transmembrane route, right? And that's going to go cell to cell from one membrane through another. All right, and then the symplastic route is a very fast route, and that symplastic route is going to go through connections between cells, these little gates between cells. And then finally, the apoplastic route is going to go into the extracellular spaces and through the cell wall. So transmembrane, not so much you have to worry about there, but symplast and apoplast, those are the ones you really want to kind of remember. So in order to increase our surface area, we have on the roots, we have what are called root hairs, and they're going to be the thing that's going to massively increase the surface area, just like we saw with the lungs and the surface area there, so that we can absorb oxygen and get rid of carbon dioxide. And then the xylem is going to be where we would look at for the endodermis, and that is going to surround the vascular tissue. And we can either go through by the symplast or the apoplast, and there is a strip here I'll show you in the next slide where the wax is going to force the water into, eventually into the apoplastic route. So here is the two routes right here, and you can see here that even though we travel through the symplastic route, eventually we will go into the apoplastic route into the xylem right here. There is the Casparian strip here, which will eventually force them into that route. Alright then, the other way that we can do this is that the water molecules themselves are going to be pulled up by this negative pressure, and that is basically a result of cohesion between the water molecules themselves, so that the water molecules stick together, and then adhesion, where they are sticking to the walls of the membrane. On the other end of it, the reason why the water goes up is something called transpirational pull, and transpirational pull is a result of a negative pressure in the leaf, and that is because the leaf is opening its stomata, and as it opens the stomata to take in air, it is going to be losing water out. So we lose water out the leaves and we gain it through the roots, and there's a pulling force called transpirational pull that does that. So between these two, we're going to be having the xylem sap, which is the primarily water, transported from the root and pulling actually stuff from the soil into the leaves. And here we can see here the water potential is going to be high at the top here, low at the bottom. All right, and as we slide through here, we're going to see the water molecules that are going to be pulled up through adhesion and cohesion, and the transpirational pull up through the leaf surface, and then there is the water leaving the plant. So the water is moving from where there's a lot of water to where there is very little water. So we're going from a high here to a low here. All right, now on the opposite end, we're going to have sugars, and those sugars are going to be coming from translocation. They're produced by the leaves, and they're going to be transported to other parts of the plant through translocation. And that is like the phloem, and that's like what you would get when you trapped a maple tree to get the syrup. You're going to get maple sap. The phloem sap is going to be something that's high in nutrients like sugar that have been produced by photosynthesis. So the source is where they're made, the sink is where they are traveling, and that could be, you know, the case of a potato, a tuber or a bulb, something like that. So the nice thing about it is we can store them during the summer, and then we can use them during the winter. So unlike the xylem, the phloem can go both up and down. Right? And of course, we're going to have a transporter here, which is going to help this concentration occur. Here's the sucrose right here being transported in. We have that same proton pump we talked about earlier, and then we have the co-transporter bringing the proton back in. So that's how we can kind of move this from one location to another, from the leaf into the phloem. So that's the active transport we just saw in the last screen. And then in angiosperms, we're going to have this positive pressure. So we have a high concentration here, and the bulk flow is basically going to push them down, and then other cells can then pick them up. So where it's negative pressure up on the vessel and xylem, it's positive pressure down on the phloem.